My name is Heather Sheaffer, and my partner is Zhao, and we'll be discussing a study done by Cayman Tan and several others at Argonne National Laboratory. The article is titled Insights into PG Binding, Conformation Change, and Dimerization of the Outer Membrane Protein A C-terminal domains from Salmonella enterica serovar typhimurium and Borrelia burgdorferi. First, it'll be helpful to start off with a little bit of background information. So when bacteria encounter the immune system cells of a host organism, an immune response is triggered by the binding of host antibodies and T cells to proteins called antigens on the bacteria's surface. Salmonella causes gastroenteritis and in some cases typhoid-like symptoms, whereas Borrelia is actually the cause of Lyme disease. Bacteria have many proteins on their surfaces, of course, but for Salmonella and Borrelia specifically, a transmembrane protein called outer membrane protein A is the primary stimulating antigen that triggers these immune responses. Outer membrane protein A is, like I said, a transmembrane protein that is anchored to the peptidoglycan, which the paper refers to as PG, in the periplasm of gram-negative bacteria. And the periplasm is simply the space between the inner and outer bacterial membranes. The basic structure of outer membrane protein A is an 8-stranded, or in some cases 16-stranded, beta barrel. And like all proteins, it has both an N-terminal domain and a C-terminal domain. So here's a picture of the outer membrane protein A. You can see its structure is a barrel that forms a pore through the membranes, and the C-terminal domain sticks outside of the outer membrane. In this study, the C-terminal domain was of particular interest. It contributes to the membrane permeability of the bacteria because the beta barrel forms a large transmembrane pore that molecules can pass through to enter and exit the cell. And as we'll see later on, the C-terminal domain actually plays a very important role in ligands binding to the outer membrane protein A. So the goal of this study was to determine the general structure of the C-terminal domain, to explore the conformation changes induced by ligand binding, and to investigate possible dimer formation, as well as determining the anchoring mechanism of the C-terminal domain to the PG within the periplasm. Then, each of these things, the structure, the dimerization, conformation change, and the PG binding of the C-terminal domain, was compared and contrasted between the two bacteria, Salmonella and Borrelia. The outer membrane proteins were lysed in a crude extract, centrifuged, filtered, and further purified with affinity chromatography and size exclusion chromatography. The monomers were then crystallized and underwent an X-ray diffraction process to further characterize their structures. It was determined that Salmonella's C-terminal domain has two crystal structures, Form 1 and Form 2. Now, Form 1 has two monomers referred to as A and B. Monomer A exists in what's known as the open form, and monomer B exists in closed form. Borrelia, on the other hand, only has one crystal structure with only one monomer, and that monomer exists in closed form. And Mimi will explain what that means.
Okay, now let's take a look at the PG binding first. So the monomer A of Salmonella outer membrane protein A, crystal form 1, is structurally similar to, almost identical to monomer B. But there are some differences that mainly result from the conformational change. For example, in the PG binding site, in monomer A, the key residue 260 is inside the binding site, and there is no small molecule located inside the binding pocket. And the sulfate anion group is located outside of the binding pocket, which make the monomer A as a open form. Compared to monomer A, the monomer B actually has the sulfate anion group located inside the binding pocket, which interact with the R260 and make it a closed form. Also, when we look at the monomer A, the key residue R260 forms a hydrogen bond with the two loops, the carboxylic acid group on the two loops, and it also forms a sulfate bridge with the residue D260, 250. for monomer B is the sulfate anion group that forms the hydrogen bond with the amide group on the main chains, and also it forms a sulfate bridge with the key residue 260. And there's another residue found inside the binding pocket too. It is the residue D245, which is more conserved, but is equivalent to D250. Overall, the D260 and R260 are the two key residues that are involved in the PG binding. During the conformational change, the open form monomer A is transformed to the closed form monomer B. So during the transition, the residue D250 actually moved from inside the binding pocket to the end of the alpha-3 helix, and the beta-2 and the alpha-3 loops become extended form of alpha-3 helix by 2 turns, which is shown on the red part of the diagram. Compared to the binding pocket of Salmonella outer membrane protein A, the outer membrane protein A of Borrelia actually is shallower and wider, and the sulfate anion only bind to the one side of the binding pocket and interact with the key residue R330. So since there is only one monomer involved in the Borrelia outer membrane protein, there is no conformational change involved. As for the dimerization, there is actually no actual dimerization exist. So dimerization is two identical tertiary protein non-covalently bind to each other. But due to the high salt concentration, the two crystal forms of the outer membrane protein of Salmonella are very unstable. So there is no real dimerization exists, but there is some possible way of forming a dimer. One is provided by the alpha-4 helix, which is shown on this diagram, and one is provided by the beta strand, which is provided on this diagram. But the beta one is very unstable. It's not as stable as the alpha-4 helix because it has a small monomer interact, and there is no hydrophobic interaction, which made it very unstable. So overall, the outer membrane protein A is also conserved in many other outer membrane proteins, suggesting a similar PG binding mechanism in other proteins as well. So, and this can be a good evidence that support a potential ligand binding mechanism, which may also enhance the dimerization of the CTD of Salmonella outer membrane protein A.
So, in summary, both Salmonella and Borrelia have overall similar monomer structures. Most of the differences between the two have to do with the structure of their PG binding pockets, in which there are different key amino acids in both Salmonella and Borrelia. And both of those key amino acids play a role in anchoring their C-terminal domains in the periplasm. Only Salmonella's outer membrane protein changes conformation from the open to the closed form when the protein binds a ligand for transport, and only Salmonella's C-terminal domain forms a dimer, which presumably helps to stabilize the structure of the outer membrane protein, although the dimer formation, like Mimi said, is relatively unstable. So not only does the C-terminal domain of outer membrane protein A play a large role in the transport of molecules through the bacterial membrane, which allows the bacteria to maintain osmotic homeostasis, but it also contributes to the structural integrity of the protein through these PG binding and dimerization mechanisms.